Shall I start? Good afternoon and a very warm welcome to everyone who has joined this virtual symposium organized by the Department of Neurovirology at NIMHANS, Bangalore. We have organized this symposium to honor Dr. Anita Desai, Professor and Head of the Department of Neurovirology, who is superannuating this month. We are here to celebrate and cherish more than 3 decades of her dedicated service to the field of virology. Following her MSc in microbiology from the University of Pune, Dr. Anita completed her PhD in neurovirology from NIMHANS under the guidance of Dr. V. Ravi as his first PhD student in 1994. She worked as a junior and then a senior scientific officer in the Department of Neurovirology and later joined as an assistant professor in the same department in February 2002. In the last 3 decades, Dr. Anita has worked and published extensively on the diagnostic and research aspects of various viral infections such as HIV, influenza, rabies, dengue, chikungunya, SARS-CoV-2 and others, especially Japanese encephalitis, which was a core area of her research for her PhD thesis. Significantly, she has led the several activities in the department funded through the National AIDS Control Organization, NACO, since 2011 and has championed the cause of HIV/AIDS. She has been a principal or a co-investigator on several research projects funded by various national and international agencies, including the ICMR, the DBT, the Wellcome Trust, Medical Research Council, and NIHR UK, and the NIH and CDC US, among several others. She has been a member of several technical expert committees constituted at the state and national levels, and a resource person for many training programs conducted at NIMHANS and elsewhere. She was invited to lead several prestigious assignments within the country and abroad, including several short-term assignments as a WHO consultant or a WHO temporary advisor, mostly in the Southeast Asian region. She has mentored several students, including 20 students pursuing their PhD in the department in the capacity of a guide or a co-guide, and seeing them placed well in their respective careers has been immensely gratifying for her. On behalf of all the faculty, staff, and students of the Department of Neurovirology at NIMHANS, I welcome you all to this symposium organized to recognize Dr. Anita's outstanding contributions to this institute and to the field of virology. This symposium is aptly titled Going Viral, Illuminating the Future Path of Virology. We are fortunate to have on board 4 excellent speakers who are stalwarts in their chosen fields and have also been closely associated with Dr. Anita Desai in a professional capacity. We are grateful to Dr. V. Ravi, Dr. Sandhya Kabra, Dr. Madhuri Thakkar, and Dr. Pragya Yadav for accepting our invitation to speak in this symposium. I extend a very warm welcome to all of them. Our director, Dr. Pratima Murthy, and our registrar, Dr. B.S. Shankar Narayan Rao, have also kindly consented to grace this event and will be joining us later. I welcome all my faculty colleagues, students, and alumni from NIMHANS who have joined virtually for the symposium. I also welcome microbiologists and virologists from several other institutes who have joined the symposium online. We request all our speakers to kindly maintain the time limit. We request all participants to type in their messages, comments, or questions for the speakers in the chat box. If time permits, we will try to address some questions at the end of the symposium. To begin, I now request my colleague, Dr. Manjunath, to introduce the first speaker of this symposium, Dr. V. Ravi.
Thank you, Dr. Rita. It is my honor to introduce our mentor, Dr. V. Ravi, popularly known as Virus Ravi for many of us, as today's first speaker. Dr. Ravi, former senior professor and head, Department of Neurovirology, Registrar and Dean, Basic Sciences, NIMHANS, Bengaluru, superannuated from NIMHANS in November 2020, He's been a member of several expert committees and continued to serve on them. Ever since starting the Department of Neurovirology in 1985, Dr. Ravi has always strived to address challenges faced with viral infections, starting from Japanese encephalitis virus, HIV, H1N1, acute encephalitis syndrome, and the most recent being COVID-19. He served as a member of the Karnataka COVID Task Force and continues to be the nodal officer for genomic confirmation of SARS-CoV-2. In April 2020, the ICMR assigned NIMHANS as a mentor institute under Dr. Ravi's leadership to facilitate the setting up of RT-PCR labs across the state of Karnataka, which has now resulted in more than 200 RT-PCR labs being established in the state. In his collective effort with Dr. Anita Desai, Dr. Ravi worked relentlessly to develop the Department of Neurovirology, which has come to the fore every time when the country has faced challenges with viral outbreaks. He has received innumerable prestigious research grants, awards, honors, and has several outstanding research publications. And whenever Virus Ravi is invited to share his knowledge and experience, he can be highly virulent and affect the young and not so young with his infectious enthusiasm, which many of us have experienced over the years. And we all know Virus Ravi can mutate and adapt quickly to tackle the challenge that lies ahead. The latest variant of Dr. Ravi serves as the head of R&D at Tata Medical and Diagnostics. Sir, we look forward to hear from you the topic, it's time to be self-reliant in diagnostics. The floor is yours, sir.
Good afternoon, and thank you, Manjunath, for a very kind introduction. My dear friend and former colleague, Dr. Anita Desai, let me at the outset thank the Virology Department for having me for this great event of celebration. Can I have the first slide, please? I will start my talk in 7 minutes. You have to share it from here?
Yes.
Yes. Full screen. Thank you, Narendra. Okay, I've titled my talk, It's Time to Be Self-Reliant in Diagnostics. I will justify why I chose this title and this topic. Before I go into the talk, I must apologize to the organizers. I'm going to take a few minutes because I'm talking about a colleague for the past 34 years, a very dear friend, beyond that, a soulmate. Dr. Anita Desai, Anita as I call her, walked in 1987 November into the virology department. I was putting some tubes for centrifugation in a Hitachi centrifuge in the old lab before we renovated. She said, "Can I meet Dr. Ravi?" I said, "Yes, you're talking to him." She said, "Dr. Gokul referred me." Dr. Gokul was a dear colleague in microbiology department. She said, "Just give me a few minutes." And then, of course, I met her, spoke to her. She was looking for a position in the department. There was one vacant. She joined as research scholar in the Department of Virology. We were only 3 people then, myself, one technician, one senior assistant, and one laboratory attendant. Since then, it has been a great journey with her. She, of course, took a break, maternity leave, between second half of 1988 and came back and joined in 1989. Very rarely in life you find a person with whom most of the things you agree. And for me, it has been Anita. 90% of the times we agreed on whatever we discussed, be it life or be it professional work. I'm showing you 4 photographs that depict her personality. The first one on the right top is with a student. She had great love for students. That is Dr. Liba Sebastian, who's now professor at Bellary. And she was also Anita's first student. That's my grand student. Anita was my first PhD student, and I must say I'm very privileged and lucky to have a student like her. She always keeps reminding me that she taught me how to be a guide, and I had to retort back saying, I taught you how to be a student. She loves traveling. You can see the pictures there. She's a super mom, a great cook, and very passionate about whatever she does. And there's one quality in her that I admire is her ability to always excel in whatever she does. She's in constant pursuit of excellence. She has been not only with me, but with the Institute in this country, contributed a lot. And you can see the photo on the bottom right, even international, University of Liverpool, WHO, and so on and so forth. So Anita, it's a privilege and a pleasure to be the first speaker in a symposium that is celebrating your contributions to the department at NIMHANS and to the larger world outside. So my congratulations for your service, felicitations and greetings, and wishing you a great post-retirement from service life. I won't say retirement. Okay. I just wrote this few lines for her, an ode of gratitude to a colleague and a dear friend. Anita, your journey's end at NIMHANS is near, a lifetime spent pursuing knowledge without fear. As head of neurovirology, you led with grace, a beacon of light in the scientific race. For 34 years, you worked alongside Ravi, that's me, your friend and guide. Together, we explored the depths of the mind to help the affected, they combined. With a passion for research and learning, you have pushed the boundaries of field, exploring the complex interactions of viruses and neurons revealed. Your work has paved the way for those who come next, a legacy of excellence and progress that's blessed. Your dedication to research is an inspiration to those who seek knowledge without hesitation. Your contributions to neurovirology are priceless and beyond any analogy. The growth of the department at NIMHANS is your legacy, and for it you stand. Your legacy lives on in the hearts of those whose lives you have touched and whose minds you chose to educate and inspire, to make them see the wonders of the human brain infections and its complexity. We wish you joy and rest in your retirement. May your future be full of enjoyment and contentment. You've left a lasting mark on those you've taught, and we'll continue to cherish the lessons you have brought. Thank you, Anita.
Thank you.
Why did I choose this topic? At Virology Department in NIMHANS, we have a range of activities: diagnostic services, outbreak investigation, and one of the important areas, apart from these two being the primary, was innovation development and validation of indigenous diagnostics. I talk about them and show them the picture in the next one. And in each one of these innovative development and validation of indigenous diagnostics, Anita was my partner, and we enjoyed working together. So also in teaching and training. Now, these were the diagnostic kits developed at NIMHANS. India's first HIV indigenous ELISA was developed in collaboration with Dr. Ravi Kumar, a very dear friend of ours, who is the chairman, managing director of Exciton Diagnostics in 1997. Then India's first commercial, in fact, I would say the world's first commercial JE IgM capture ELISA was again developed in collaboration with Exciton. And the third was molecular assays, which we developed, of which Anita was part of the whole development and validation, and so was Rita. In fact, Rita joined the department as a project officer in that. Apart from these, one of the things that NIMHANS takes pride is an assessment of quality of HIV kits that are used. Dr. Sandhya Kabra, who was speaking after me, was a prime mover of this, of establishing a reference laboratory on quality for the country, which even today tests every batch that is supplied to the National AIDS Control Program. This taught all of us a great deal on diagnostics and how to maintain quality. So with this background, I'm moving to my presentation. I hope to complete exactly at 2:30. What I'm going to do is three things. First of all, address why do we need to be self-reliant in diagnostics? What are the hurdles for being self-reliant in diagnostics? Where do we begin and how do we navigate? And end with a summary. Why be self-reliant in diagnostics? There are 6 points I want to put as answers to this question. Sheer economics. It's profitable and it's $1 billion industry. In the next slide, I will tell you how big the industry is. With COVID, we learned a very, very important, maybe initially a very bitter lesson, that we were wholly and solely reliant on imports. It was pandemic time and every country was looking after its needs. So whatever it was giving to other countries was at a premium. So we burnt a lot of money. I don't think we should be doing that in future. We have some peculiar needs and also we have some peculiar problems. Let me come to the needs first. Accessibility and affordability are an issue in diagnostics. For a country that has been reliant on imports, 90% of it, this is a very, very, challenging to address it. And also our needs are not like rest of the world. India, I always tell people, is not one country in terms of population, it's 30 countries. Geographies differ, terrains differ, and also the cultures are different. Health-seeking behavior is different in different parts of the country. So I think a diagnostic made in the Western world will not straight away be adopted or can be adopted as it is elsewhere. And also we have some peculiar pathogens and combinations of pathogens which Western kits do not address. I'll give you an example. I don't want to name companies, but there are companies which offer febrile illness panel, encephalitis panel as molecular tests. But in one panel, you will not get all the pathogens in India. For the company, it is profitable because you will have to, Japanese encephalitis will be in one panel, scrub typhus will be in another panel, dengue will be in another panel. So you end up buying 3 panels and the cost will be something around 5,000 to 10,000 rupees. Nobody has put a panel together for our needs. We have to solve our problems and we have to address our needs. I think also being self-reliant in diagnostics brings a great deal of accountability. You can catch people by their neck, not by their hair, because people like me in the industry don't have hair on their head. So it's better to catch them by their neck. And also, I think it's a great challenge. And it's national pride that we should be able to develop our diagnostics. We have the brain power, we have the infrastructure, we have one of the lowest manufacturing costs that any country can have, probably even less than China. Then why are we bleeding dollars? I don't know. This is something that has always appealed to me. Here is the justification. At present, in the year 2022, the diagnostic industry is worth $10 billion, and it's projected to double in the next 3 years. By 2026, it will be $20 billion. Just for you to understand, $10 billion is equivalent to 82,679 crores. It's almost, you can say, 1 lakh crores, because many of them are not accounted. And it's growing at a very fast rate. Compared to population, urbanization, you can see the highest growth rate, it's about 14%. It's for diagnostics. In the next 3 years, that's the growth rate we are looking at. And in them, of course, pathology, as they call it in diagnostics, is basically, biochemistry, hematology, infectious disease. The ecosystem has expanded, but I will give you a minute's challenge. I'm not going to waste that minute and ask you to identify one company in that is Indian and that's a leader. You will not find it. Thermo Fisher, Roche, Abbott. These are all the companies that we are absolutely reliant on. Why? I don't know why, but it's time to ask the question why. Look at what is happening within the country. The government is taking several new initiatives. All of them are on this slide. 3 diagnostic services under National Health Mission. National Health Essential Diagnostics List is going to be implemented. I'm sure all of you are aware about the WHO's Essential Diagnostic List. I happen to be in the expert group that formulated the Essential Diagnostic List for India. So all of them are going to be available at various levels, and they have been assigned different types of tests for each level we are talking, right from PHC to Taluk to district and above. National programs are reliant on diagnostics, huge programs. TB program, Dr. Sandhya will talk to you about hepatitis program. We don't have a program for cervical cancer, but it's on the anvil, it's going to come. All big killers, all for diseases of great public health relevance. And also, there are models of government partnering with private players, the PPP model, so to say. Beyond all this, there is a growing culture of people getting themselves tested for wellness, especially in urban cities. They spend huge loads of money. Now, as I said, COVID was a great teacher. It taught us many things. Look at what happened during COVID. Prior to COVID in 2019, the market size and the availability was so tiny. You can see the green bar there. And then, of course, during COVID in 2020, it grew several hundredfold, and both in terms of sales as well as in revenue. What are the hurdles to the path of self-reliance? I think there are several hurdles. The first two are very important. The mindset and the perception. What do I mean by mindset? There is a very firm belief in, at least in national programs, and for their very valid reasons, probably they are right, that closed molecular systems or immunosystems are probably best suited because they can be, taken to the last mile. For example, a viral load assay or a TB assay. If you make an kit that can be used on open PCR. In the last 2 years, I've learned great lessons. People have told me, no, thank you, we don't want it in the national program. I don't know why. I asked the question, we created 6,000 COVID labs. COVID is going or gone. What is going to happen to all that infrastructure? A COVID PCR lab costed 1 crore. What happens to all the investment? Is it going to rust because COVID is gone? Can't we redeploy it for diseases that are much more serious and have been around for decades, which kill millions of people? TB, human papillomavirus, hepatitis, HIV. I don't know. I think this is something, this is mindset. Perception, this have been fighting for the last 3 decades. Indigenous diagnostics, the equation is cheap and poor quality. Probably there are reasons to arrive at this equation, but this equation has to be changed. We can make very good qualities. And why should it be cheap? Quality and affordability don't go very, low cost and quality don't go hand in hand. They are opposites. There is a price for quality. So you'll have to spend money. But you don't have to spend in dollars what you're spending now. Unrealistic expectations. Whenever I talk to people and said we have developed an assay in the last 2 years, they said, do you have FDA approval? Excuse me? Go and get FDA approval. It takes 1.5 years. When it's a non-American company, you go down to the bottom of the applications because they don't want you to cut into their business. They take their own sweet time. You want WHO emergency use authorization, you have to go in a queue. And we are at the last step of obtaining emergency use listing. One of the first kits in the world for variant detection, PCR, separate category. No other Indian company has reached that stage. Scalability is a big hurdle, both in terms of the geographies we will have to provide kits in this country. If you go to Northeast, the terrain is very difficult. The infrastructure is still not what it is in the mainland. So there are difficulties, and also accessing international markets because of regulatory hurdles. Biggest hurdle, I would say, as a scientist I have faced is lack of well-characterized samples. Dr. Sandhya, thank you very much. I think we have shown an example in the NRL on Q that we can build a good biobank of well-characterized samples. I think that is needed. I know I respect ethics. I've been in various ethics committees, but even ethics is evolving, saying that dustbin samples, if properly worded and consent obtained, can be used for developing newer tests. Regulatory challenges, and of course, one of the reasons for the equation which I showed you on the top is fly-by-night operators. They open a shop, sell some kits, make money and shut shop, and they give 2 hoots for quality. There are also people like that. Regulatory is a very big challenge. There are no proper protocols, very ad hoc approach to validations. It takes 120 days to get a license, yes or no, after providing all the data to the regulator. And they are absolutely in their own world, not in tune with international standards. So no wonder it's very difficult to get FDA approvals. I just spent the last few minutes on talking about Tata MD. What are we trying to solve? 2 things we're trying to solve. We're taking everything closer to the patient. Diagnostics closer to the patient, make it affordable. And also healthcare delivery closer to the patient, which I will not talk today because it is basically, we believe there is tremendous innovative potential in the country. We want to leverage the low cost of manufacturing and make kits available for this country and rest of the world. We have a unique molecular diagnostics platform, which has been an outcome of research for the last 15 years of me and my colleagues, including Dr. Anita was part of it, where we can design now primers and probes for molecular tests in a matter of a week. And that's an example of the OmiSure, the world's first variant detection SARS-CoV. It was announced on November 26th. We decided to make a kit on December 1st. December 7th, we had designed primers and probes. 20th, we got them imported. 23rd, we filed a patent. 26th, we sent it to ICMR for validation. 30th, we got the report. 1st January, I developed COVID, and mine was the first clinical sample to be tested. And 4th January, we got license. It is a test that is equivalent. It uses two strategies, an S-gene dropout and an S-gene mutation-specific amplification for Omicron, and also it amplifies all other variants. It is equivalent 99.5 to 100% equivalent to genomic sequencing. And it is the only test, this is the comparative chart, it's the only test that can detect Omicron specifically and also other variants. We have a variant watch platform. Every week, twice, we look at the sequences from the global database and see its impact on the performance of our primers, probes, and the assay. And I'm happy to tell you, till today, every sublineage of Omicron is being detected. So that now that SARS-CoV is on way out, we are now expanding into non-COVID diseases, tuberculosis, HIV, HBV, HCV, SARI flu, dengue, and cervical cancer. We are also into partnering with firms to bring in novel and equipments that are affordable and that can do the test fast and that can be point of care. Now, these are the analyzers that we have, a very smart portable PCR, point of care, TB diagnostic tests. At Tata MD, we don't believe in selling products. We believe in offering solutions. So we're coming up with a digital X-ray for TB screening, a specimen collection universal medium, which we will be released shortly for all molecular tests, that hopefully will obviate the need for nucleic acid extraction. We offer testing on mobile vans that are NABL accredited, and also drug-resistant testing. All this for tuberculosis. For HPV, we come up with a self-collection brush, a reconstitution medium. The brush is dry, it can be posted. And then, of course, testing for all high-risk pathogens. So this is the kind of challenges I've taken up post-retirement. I'll end by telling there is compelling evidence to suggest we need to move self-reliance in diagnostics. I gave you economic arguments that is profitable. You can be a global leader in this. You can address needs and challenges specific to our country, and above all, be accountable. And to succeed this, it is not only the industry, it requires a lot of support from academia, from hospitals, from regulators. We need to change our mindset and perceptions. And the ecosystem need to change for validations, regulatory, etc. Lastly, we have the brain power, the muscle power, and we should harness the low manufacturing capability of India to cater to global markets. Thank you very much.
Thanks, Dr. Ravi, for the excellent presentation of reflecting the past and giving a glory of the potential of Indian diagnostic industry. So I move on to the next speaker, Dr. Sandhya Kabra. She is currently the Deputy Commissioner and Additional Director, National Center for Disease Control, and is the technical lead for the National Viral Hepatitis Control and Surveillance Program. She has done stellar work in the National AIDS Control Program, where she was involved in lab strengthening and also building quality systems in the HIV network of labs as well as the blood safety section. And she has contributed immensely to developing of quality assurance and legal framework related to quality assurance in India. And she has over 25 years experience in diagnostic services, program development, policy formulation. And she will be speaking on India's response to combating hepatitis. Over to Dr. Sandhya.
Thank you very much. Good afternoon, everyone.
Good afternoon.
Thank you to NIMANS to have thought us worthy of making this presentation. And we have, at least in my life, I have truly gone viral. We started with a career where, we were taught that virology was a very small field. You just needed to pass a paper. And we did our MD looking at diagnostics and thinking we probably won't be using them in virology. Those were different times. And 30 years down the line, we can surely say I've served 8 years in the National AIDS Control Program and nearly 5 years here with the viral hepatitis program. So one doesn't know what's going to happen tomorrow. And today I can say that we've truly gone viral after COVID, after so many challenges that we are seeing with viral diseases. But I'll stick to the theme of the viral hepatitis program because I think it's important for us as scientists, as microbiologists, as people who deal with the diagnostics to understand that our roles are not limited to the bench and inside the laboratories, but they have long repercussions and implications on the lives of people in the public health domain. So I'll just request for my presentation to be shared, or should I share it myself?
Yeah, Dr. Shandre, you can try and share. Because that will be easier for you to handle the slides. Yeah. Otherwise, we can always get it.
Be the co-host.
Yeah.
Co-host.
Yeah.
Yeah. Yes.
Yeah. So is it visible? Can you put it on the view?
One moment, madam.
Yeah.
See yes.
Yes.
Narendra, see yes.
Not getting that screen.
Narendra, on the screen. Yes. See yes then.
Yes.
Visible now?
Now you can share.
Yes, it's coming up.
Yeah.
There it
Great.
is.
Thank you. So I'll be talking about India's response towards combating viral hepatitis. The next slide. So the outline of my presentation is I'm just going to talk of the burden of the disease both globally and in India. And viral hepatitis elimination targets as set by the WHO and India's response to these targets to combat hepatitis. Next slide. So a little bit on the burden of the disease. Next slide.
Just 1 minute. Just 1 minute.
1 minute.
Just 1 minute, Dr. Shandre.
Exit screen. Exit full screen.
Yeah. Okay, can I go ahead?
Yes, madam, please.
Yeah. Okay, so the burden of chronic hepatitis B based on the hepatitis B surface antigen positivity in the Southeast Asian region is nearly 60 million people are estimated. Next slide. And for hepatitis C, we have nearly 30 million people, 10 million people estimated with the disease. Now, the important thing to note is that we had actually no estimations in the country. So we were just working on the WHO estimates till the program was even launched. And then we... is there a problem or can I continue? Am I audible? Am I?
See.
Just a minute, Sandhya. There are two tracks that are clashing. Just give us a minute.
Okay, no problem.
Yes, Sanjay, you can continue now.
Okay, thank you. So as I said, that we didn't have any data with regards to the disease seroprevalence or the burden, and it was all dependent on a couple of research studies published, which was a bit skewed. And then we got an opportunity to retrospectively analyze, the dried blood spot specimens which were lying in the country, which were collected in 15-16, which we managed to actually validate the dried blood spot methodology and then do the testing. So we had a rough estimate that we could get retrospectively, which said that we have a seroprevalence of around a little less than 1% for hepatitis B and seroprevalence of 0.32% of hepatitis C. This is the national seroprevalence. Next slide, please. And we are committed towards the Sustainable Developmental Goal Target 3.3, which is to combat viral hepatitis. Actually, the roadmap to elimination is a huge task, and it proposes 65% reduction of deaths and 90% reductions in new infections. Next slide. So if this is a little bit of the international norm for how would we declare elimination. So there is incidence has to be reduced by 95%, mortality has to be reduced by 65%, and we have targets for mother to child elimination, both for hepatitis B and C. There are separate targets. Next slide, please. And of course, we are a country where we have universal hepatitis B vaccine birth dose as well as the others. And there is a target for us on there. We are pretty much in line over there. And we collaborate very closely for the harm reduction piece, which is there should be 0% unsafe injections, 100% blood safety, and there must be a needle exchange of at least 300 needles per syringe per person who injects drugs per year. And this target, actually, we are piggybacking with NACO, and we are achieving this. But yes, injection safety still remains a huge concern for our country, especially in the Northern Belt. Next slide. Next slide. So the program was launched in 2018, July, and we rolled out with the treatment of hepatitis C free of cost, and expanded this in 2019, February, to include lifelong treatment for hepatitis B. And we also launched our helpline and our MIS portal, which is the Management Information System, which is a paperless portal on the program website. Next slide. So in the process, ours was a little unique process as we conceptualized this program not as a vertical program, but as a program within the health systems. And we involved all stakeholders, not only administrative and political leadership, but technical experts and community were very closely associated with drafting of this program. And finally, we managed to get the approval of the program. The good thing about this program, I think Dr. Ravi was talking a lot of national interest, national revenue, and investment. So this is a 100% domestically funded program where sustainability is not an issue, because we are committed as a country to support the program throughout. Next slide. So a little bit about the program. So what does the program offer? The program is under the UHC, the Universal Health Coverage umbrella for viral hepatitis. We want to ensure access, address quality, and ensure financing, which we have already done through the domestic funding. And if you look at the blue piece, which is the prevention, diagnosis, management, which includes treatment and monitoring, and capacity building, which includes a lot of training, this is what the program finances and budgets for and provides. Besides this, we closely integrate with a couple of other existing programs. So we are not a vertical. We need to closely integrate with the Reproductive and Child Health Division, the Immunization Division, the National AIDS Control Program, Safety of Blood and Blood Products, and the Surveillance Program. A little bit on the safety of blood and blood products. We are aware that as a TTIs, hepatitis B and C are two important markers. And we are promoting that all donors who come out to be screened positive must be referred into the arm of the program for further confirmation and management. And the immunization program supports us by providing us vaccines for the baby, which is the 0 dose, and all the other doses for hepatitis B as a preventive mode, and vaccination for all healthcare workers in the country. And in the surveillance program, I'll be talking a bit more about the rest of the three components a little later. Next slide, please. So this is our organizational structure. At the national level, we have a technical team and an administrative team, same at the state structure, district structure, and we do want to go below district level for our, we really wish to reach the last man by going to the community health center, primary health center, and the health and wellness center, and try that at least screening happens at our last lab. And then wherever there is a medically qualified doctor, prescription can be given and treatment can be provided from there. Next slide, please. So in diagnosis and management of hepatitis B and C, we use the rapid screening test for hepatitis B and C both. And we screen all who have a perception of risk by using serum, plasma, or whole blood specimens. All positive specimens have to be confirmed by a molecular testing for especially for hepatitis C, and we do require a quantitative HBV DNA estimation to decide on the need and the type of therapy for the hepatitis B patient. So this is a little bit just as a passing slide. I do want people to be aware that we are offering this treatment, which is available world over, but we offer it free of cost in a public health program, and we are probably one of the largest programs of the world offering this. So we provide for treatment of uncomplicated patients. We provide sofosbuvir and daclitazide. It is only a 84-day treatment, which is hardly 3 months. And if you can be rid of a disease like hepatitis C and the complications, it's a great thing. And we also offer a treatment to patients of complicated, which are cirrhosis, both compensated and decompensated. These are the regimens. And same way, we provide for management of hepatitis B. In uncomplicated cases, we use TDF. And in complicated cases, depending on various factors, we use entecavir, either in the strength of 0.5 milligrams or 1 milligram. Next slide, please. So this is a little bit on the integration. In this, basically, how have we integrated with the existing reproductive and child health program arm? All pregnant women. So the program recommends universal screening of all pregnant women across the country with hepatitis B. And if diagnosed as hepatitis B surface antigen positive, this woman is labeled as a high-risk pregnancy. Everything that the RCH offers for a high-risk woman is provided to this lady. She is then referred for institutional delivery to a referral center. And in this referral center, we provide for hepatitis B 0 dose through the immunization program. And the National Viral Hepatitis Control Program provides for the hepatitis B immunoglobulin. And both these together, if administered within 24 hours in public health terms, provide near protection to babies from mother to child transmission. Next slide, please. So with the AIDS control program, you're all aware of the AIDS control program. We are promoting for testing of all clients at ICTCs and all PLHIV at ART to be screened necessarily for hepatitis B and hepatitis C. And anyone who is found to be positive, we have to link them for further management with the hepatitis program. Here, we provide for capacity building and we provide for the screening kits. However, we want to use the same equipment, we want to use the same machinery, manpower, and existing infrastructure in the health systems. In the high-risk groups, as you're aware, the targeted intervention sites of the NSCP hold the targeted intervention or the high-risk groups, and including opioid substitution therapy centers, where we have a two-pronged approach. Either we go in a CAM-based mode to these TI sites and we screen, and whoever is found positive, we link them to treatment. And for hepatitis B, if these high-risk groups are found to be hepatitis B surface antigen negative, we propose for vaccination of these three, and we want to do a full vaccination for hepatitis B for these people so that in future they don't get infected. And we also offer to these people, if they do not want to be tested in a camp mode, they can come to any one of our facilities. And here again, we provide for the screening kits, we provide for the hepatitis B vaccine and capacity building. Next slide. So this is the integration with the ongoing surveillance. Many of you might be a part of this. So we are, of course, having our own passive surveillance through the control program. We also have a surveillance program separately, which is a central sector scheme run under the NCDC. We are we have already piggybacked for biomarkers of hepatitis B and C on the HIV sentinel surveillance. One round is over. We are on to the second round. In the National Family Health Survey also, finally, we have been allotted official space. So NFHS 4, we sort of bulldozed our way into doing the retrospective sampling. NFHS 5, we managed, and NFHS 6, officially, we are into this. And we will now have data on population-based surveys. Next slide, please. We also closely integrate with many other divisions, departments, and programs. Here, I would like to have a special mention on the cancer control program, which is actually a non-communicable disease, but primary hepatocellular carcinoma very often occurs if you have infection with hepatitis B and C. And now our focus as the program expands is not only going to remain on screening, early screening, diagnosis, and management, but also to look at our cancer registries and see how this number can be reduced by doing early management for these diseases. Next slide, please. So this is our current status. We've covered 100% of the districts, and we've covered 98% of the districts, even though the program has to be rolled out in all districts. We have 893 treatment sites established. So we are a fairly new program. We were essentially rolled out in 2019, then COVID has come. So we've still managed to scale up considerably. We've screened more than 50 million people as of December 2022, and 0.19 million people have been initiated on treatment or completed treatment. And we have trained nearly 2,200 master trainers. And the dark blue color on the right side of the map, as you can see, is the states in which we have decentralized below district level to offer services to the system. Next slide, please. Thank you very much.
We are very thankful to Dr. Sankhya for introducing us to the existing preventive measures, management measures for viral hepatitis, and illuminating the path for combating viral hepatitis for the country in the future. Thank you very much, madam. And we sincerely apologize for the technical glitch during your presentation. And with that, it's my privilege to introduce the next distinguished speaker. Before that, I would like to mention to our viewers, please leave your questions and comments on the YouTube chat box, and we will forward them to the respective speakers to get back to you. And I would like to introduce our next distinguished speaker, Dr. Madhuri Thakkar, who is currently serving as Scientist G at National AIDS Research Institute. Dr. Madhuri has been at NARI for more than 3 decades in the division of serology and immunology. Her areas of research interest include immunological correlates of protection in HIV-infected patients with absence of disease progression, patients with progressive HIV infection, and patients on treatment. And she's been putting in a lot of hard work to establish long-term non-progressive cohorts to study the correlates of protection, which has been one of her main areas of research. In particular, she has conducted studies on HIV-specific antibodies such as ADCC mediating IgG antibodies, innate immune effector cells such as natural killer cells, natural killer T cells, and HIV-specific CD4 and CD8 T cells. Mucosal immune response, both innate and HIV-specific, to understand the mechanism of HIV acquisition and resistance. Another important thrust area for Dr. Madhuri has been quality control in HIV laboratories. She has been in charge of Apex Laboratory to support National AIDS Control Program. Under her leadership, NARI has been proficiency provider for external quality assurance in HIV serology and CD4 count estimation in more than 254 laboratories under the National AIDS Control Program. She has received ICMR Excellent Research Output Award, Fogarty Scholarship for training on mucosal studies in the US, and she also has received a research fellowship at Pasteur Institute under the program title Immunoregulatory Mechanism in Mother-to-Child HIV Infection. She has several publications in peer-reviewed international journals, and I herewith welcome Dr. Madhuri to talk on her topic, HIV latency, a roadblock in HIV cure.
Thank you. Thank you for this kind introduction. Good afternoon to all of you. I feel privileged to talk in the symposium which has been organized to recognize the immense contribution given by my beloved friend Dr. Anita Desai to the field of virology in the country. I would now like to share my screen. Can I do it now?
Yeah. Yes, you can, Dr. Madhuri.
Focus.
Is it visible?
Yes.
Okay. So I'm going to speak on HIV latency, a roadblock in HIV cure. We all know that HIV is the most devastating pandemic in this century. This is a unique disease where the infectious agent infects the cells of the immune system, making the infected individual immunodeficient without any underlying cause, but because of the infection with HIV. The good news in this is with the advent of ART, the HIV infection has become a chronic manageable disease now. But the bad news is, although ART potently inhibit HIV replication, they do not eradicate the virus. And that's why the complete cure from HIV is impossible as of today. Because of this, there is a requirement of complete adherence to lifelong ART to keep the reservoirs away from activation, and which because of that, the people on treatment experience various side effects of ART, such as chronic inflammation and occurrence of various NCDs. What are these HIV reservoirs? So HIV is known to persist in a small pool of cells that harbor both integrated and replication competent HIV genome. And these cells do not produce the virus unless they get activated. So we can define the latently infected cells as the cells harboring integrated and intact provirus that do not actively produce infectious virion but are ready. When they get activated or stimulated, they have the capacity to produce the infectious virus. It has been thought that there are two types of reservoirs. There could be two types of reservoirs. One is maintained because of the residual virus replication during ART, which can be attributed to the suboptimal diffusion of ART in lymphoid tissues. The other most important type of reservoir is the viral reservoir that contributes to the HIV persistence. And these are the latently infected cells that persist for decades in HIV infected individuals receiving ART. It is known that the reservoirs are established very, very early in the infection, as early as 10 days. So once the HIV person gets infected with HIV, the reservoirs are established unless the ART is initiated along with the infection very early. Actually, these cells are extremely rare. It has been shown that around only 1 in 1 million restrict CD4 cells is the HIV reservoir. But since it has a capacity to long live with an estimate of half-life of 44 months, that indicates that ART alone will not irradiate the pool of latently infected cells in a lifetime. And that's why it is becoming the most important roadblock in HIV cure. The resting memory CD4 cells are the major cellular reservoir because the viral DNA has been found in all the different subsets of resting memory CD4 cells, from primitive T cells, different types of central memory T cells, central memory, transitional memory, and effector memory cells, and also in stem cell memory T cells. The lymphoid tissues have shown the presence of the HIV reservoir in CD4 positive Th17 cells and T follicular CD4 cells. There are other cell types also who can have the HIV genome which is silenced. These are monocytes, tissue resident macrophages, follicular dendritic cells, hemopoietic progenitor cells, and astrocytes. The tissue also has reservoirs, and these tissues primary from gut and lymph node, and at lower frequencies, the HIV reservoir has been seen in spleen, liver, lung, CNS, and bone marrow. How the latent reservoirs are established? They are established by two different pathways. One is the routine, like infection of CD4 cell. When it gets activated, virus enters, the DNA, RNA is converted into DNA and gets integrated into the cell genome. There is a HIV replication, the death of the activated infected cell. Of this, some percentage of cells return to quiescence stage, and these are become latently infected memory CD4 positive cells, which remain at a quiescent stage, by silencing the HIV promoter till they get activated. The other two types of the establishment of latent reservoir is without T cell activation, where the CD4 T cell, which is G0 memory state, or T memory cells, they get infected directly by the HIV, and since they are not multiplying, they are not activated, the HIV genome gets integrated into the cell genome and remain there till they get activated. How the HIV reservoir persists during ART? There are three different principal phenomenon that have been proposed. One is self-renewal and long-term survival. So the stem cell memory, central memory cells, they have a capacity to self-renew and remain long-term in the their site. And that's why the persistence of HIV reservoir is maintained. When the cells like effector memory T cell, when they get proliferated, they take their genome along with that HIV genome to the progeny of proliferated cells, and the HIV reservoir is maintained. And the other important phenomenon is, since the cells like T follicular helper cell are in B cell follicle, they are not, they are protected from the killing of infected and virus protein expressing cells by CTL, and that's how they persist in the human body as a latent reservoir. The different strategies for removing the latent reservoirs have been tried. The one most important and tried widely is known as shock and kill strategy, where the drugs are used to activate the gene transcription, that is called as shock. So this is an HIV reservoir with the LRA, that is latency reversal agents. The cell get activated and it starts its transcription, and that's how proteins are assembled together to form virions, and the virions are out of the cell. The cell become infected and express the virus protein on the cell, which are cleared by the immune mediated clearance or virus mediated cytotoxicity. There are different latency reversal agents tried, and such as epigenetic modification in enzyme inhibitors, HDA, CI, histone methyl transferase inhibitors, and DNA methylation inhibitors. Different cytokines and receptor agonists, cellular signaling modulators such as protein PKC receptor against prostatin and bryostatin. Some of these LRAs were tested in the clinical trial, but overall it has been found that the LRAs can reactivate the expression of HIV, but they cannot eliminate the latently, means activated infection cell, or even they have no effect on the size of the HIV reservoir. Hence, now people are trying to use the strategies for immune modulation along with LRAs, and to see what kind of effect it give when it is used in combination. The other new strategy which has been thought over is block and lock strategy, where as name suggests, that the transcription of HIV genome is blocked and locked in such a way that it remains latent and there will not be viral rebound possible by possible complete silencing of the remission of HIV. However, this strategy will require the compounds which have a capability to prevent the reactivation of latent HIV, and then the cure of HIV can be attained. one of the most attractive targets to suppress HIV and prevent its rebound is the inhibitor of that protein by using DCA. But there are some real challenges for this deep latency strategy. One is how to effectively induce the silencing to prevent the viral rebound. And second, once that deep silencing is induced, how to maintain the complete silence state in reservoirs for a very long time. So this figure shows the proposed use of both LIAs, that is latency reagents, and the epigenetically modified compounds, which can have a modification of local chromatin environment in HIV LTR. So if you use those that combination, it is thought that once the viral load is undetectable or at the detection limit by ART, when this compound will be used, there will be very low or no rebound when the ART is discontinued. The drugs that can induce such modifications are being invented, and lately the bioinformatic approach is also being used to design such type of drugs. Now I will switch gear to a little bit, and I will give some glimpses of our studies, what we are conducting on reduction in latent reservoirs. We have used different types of LRA. We think that HIV antigens can also be used as an LRA. It has two effects. One is they can activate the silent infection, and the second one is, since they are HIV antigen, they can act as an immune stimulator to the immune response against HIV also. We have we are working on two important mechanisms that are involved in killing of infected cells. Those are NK cell mediated ADCC and HIV specific cytotoxic T cell mediated cytotoxicity, which we think that these mechanisms will act on the activated or stimulated latent reservoirs, which are expressing the viral proteins on their surface. The first one, what we did was to see whether envelope, if we use different types of HIV antigens, whether they can stimulate the CD4 in HIV infected CD4 cells to express P24, indicating that they are now actively in multiplication of virus. So we did that, and we found out that out of these GAG, envelope, Pol, NEF, fake and PPD are the positive control, but envelope showed very good reactivation of infected cells both in ART naive individuals and virally suppressed patients on ART. So in further experiment, we decided to use envelope as an LRA. And we developed a latency reduction assay. I'm not going to go into the details of the assay, but just to say that we used a sorted cells resting memory, that is a target, that is a HIV reservoirs from HIV infected individuals, and then mix them with either NK cells or HIV specific autologous CD4 cells. They were reactivated with envelope, and which the reactivation was assessed by cellular P24 expression, and then either ADCC IgG antibodies were added, or the CTL added with the fat sorted autologous resting memory cells were checked for their effect on the both on reduction in the latent reservoir by assessing the intracellular P24 expression after addition of antibodies or CTLs. And we simultaneously, we also check the functionality of NK cells as well as HIV specific CTL. Now, first I'll give you some glimpses of ADCC studies. So what we did first, we used the plasma of the infected individuals which have shown ADCC, and we purified the ADCC mediating antibodies, and we used these in latency reduction assay. And what we have found out, when the percentage of P24 CD45, that is memory cells activated, found out that when we added ADCC antibodies, then only there was a significant reduction in P24 expressing cells. as compared to when there were no antibodies, or even the antibodies which are added, which didn't have ADCC activity, and HIV negative antibodies. Simultaneously, we saw that when the IgG having ADCC and capacity were added, there was a increase, significantly higher increase in the functionality of NK cell as measured by CD107A, that is cytotoxicity, and gamma interferon secretion. Then we, as we know that the antibodies from the HIV infected individuals are ART, those ADCC antibodies have are functionally less competent. We thought that it will be good if we can generate ADCC mediating antibodies and see that if they have the capacity to lyse the reactivated reservoir, this could be a new avenue to act as an immunotherapy. So we purify the envelope specific memory B cells from HIV infected individual and put them in long-term culture. Then the antibodies secreted by these culture, we saw whether they have any ability to lyse the HIV infected activated latent reservoir, and we saw that the percent P24 expressing cells was decreased when we added the envelope, ADCC antibodies from generated from memory B cells of HIV infected individual who have shown ADCC antibody activity in their plasma, whereas the other individuals which did not have this activity, the memory B cells of these individuals showed ADCC activity, secrete antibody which are having ADCC activity, but which was lower than the other counterpart. The similar results were seen when NK cell activation was observed, which was more with antibodies from individuals showing ADCC activity. And when we used, this just to show that the HIV uninfected individual did not show any activity, giving the specificity to the ADCC generated from individuals with HIV ADCC activity. The CD8 CTLs, we all know that these are the primary cells which have the great importance in viral infections, where these cells kill the autologous cells, which are target cells which express the viral antigens on its surface. But in HIV infection, it is different scenario. CD8 CTL, although they are important, but they are exhausted because of the continuous chronic inflammation and stimulation. And that's why we wanted to see whether these cells have shown any exhaustion shown in the experiments. So what we did, we CD8 cells from HIV infected individuals on ART, 8 such individuals, we stained the PBMCs, and we found out that the CD8 cells of these infected PBM individual, when they were stimulated with envelope, their exhaustion was increased. It was significantly higher as compared to unstimulated PBMC. But when we did separated out the PD1 expressing CD8 and PD1 not expressing CD8, we found out that the envelope stimulated PBMCs which were not expressing PD1 had higher functionality. So that's why we thought that PD1 has a role in exhaustion and also in attaining the functionality to the CD8 cells. So we decided to use the PD1 blockade and do the latency reduction assay and see whether they have any impact on the lysis of the reactivated reservoir. We added PD1 antibodies to the cells, and when we added that the PD1 antibodies, the functionality of the CD8 cell was rescued, and it was considerably higher than the CD8 cell without PD1 blockade, unstimulated and envelope stimulated memory B cells. And when we saw analyze their ability to lyse the reactivated memory cells, it was observed that when there was a PD-1 blockade, the decrease in the reactivated memory cells was higher as compared to the cells where the PD-1 blockade was not obtained. So it was shown clear that the addition of PD-1 antibodies, they rescued the CTL functionality and increased their ability to kill reactivated latent reservoir. So to conclude, there is still a lot to be done to remove this roadblock. We have to identify good combinations of LRAs, immune stimulators, and as well as checkpoint blockers, as we have seen that PD-1 blocker could be useful. We must have standardized but easy to do assays to determine the latency reversal and killing. We also should have assays to measure the pool of latent reservoirs. And we must have the availability of primary samples from HIV infected individuals with early ART initiation to really see whether there is a lysis of the reactivated cells in these individuals if there is an early early ART is initiated. And I feel that all these efforts will bring us closer to the HIV cure. To acknowledge, I would like to acknowledge ICMR and DBT for funding the research, and staff and my students for conducting this research, Dr. Ashwini Shete, Dr. Poonam Suryavanshi, Dr. Jayashri Dande, and Mrs. Shubhangi Bichare, and at the last but not the least, the study participant. Thank you very much.
Thank you, Dr. Madhuri, for the wonderful insights on HIV latency and the progress so far. Our next speaker is Dr. Pragya Yadav. Dr. Pragya Yadav is a scientist, F and group leader, maximum containment facility at National Institute of Virology, Pune. She is known for her stellar work in the COVID response of India, and also for developing part of the team that developed the indigenous vaccine and demonstrating the efficacy of Covaxin against different SARS variants. So she's also worked extensively on development of kits and the basic biological aspects of multiple India-specific diseases like KFD, etc. And she has more than 290 research publications, which have almost 8,400 citations. And she'll be speaking on the role of genomics in preparedness and tackling of viral outbreaks. Over to Dr. Pragya.
Thank you so much for kind introductions, and it's really great privilege to present here in front of Dr. Ravi sir, who we all always praise and inspiration for us. And I was searching some photos for Dr. Anita Desai. She's an inspiration for all young virologists. I should put it very straight. And I found this very interesting picture, which just explained 3 generations in one photo and one mentor to another. So I found this legacy of NIMHANS, which Dr. Ravi has started going on. And I think we have a great source of knowledge, which coming from this institution. And the messages which he always given, I used to attend very early conferences when I was a student, and still he inspired in his each species and speech and give a new direction. So I can say that when the guide is so great, so the students sometimes just become better than guide. That's what he said in the beginning. And and we always admire, and we from ICMR National Institute of Virology, I send the best wishes from virology family to Dr. Anita for her future career. I don't think any virology ever retired, so she is not going to retire, but I think she is going to use her best mind after this superannuations and serve the country and international community. And we look forward to work further with her in the second inning of her tenure. So with this, I dedicate my presentation to her today for her dedicated services in the field of virology. And I always read a lot of papers which come from any of the country. And I read recently some of the Dr. Anita's paper, and they are really remarkable studies which she has done. And I really admire the whole team which work, and they're very fortunate to work with such people who has such a great depth of knowledge in virology. With this, I share some of my thought on the role of genomics in coming preparedness for the virology outbreaks and pandemic. As we all know, and Dr. Ravi sir had highlighted in the beginning the strength and the weaknesses which we observed during the COVID pandemic. And one of the strengths which we gained through the pandemic is the genomic surveillance. And we all know that during the pandemic, even a small child now know what is sequencing, why it is required. And we all understand that its role in the clinical epidemiology and multi-source data, genomic data for pathogens, either with any of the potentials for risk assessment, development of vaccines, therapeutics, diagnosis, and also a decision on public health and social measures. During the pandemic, we have seen a very clear-cut observation, emergence of new variant of concern, and based on that, which countries will be locked down, which border will be closed, which country resident will not come to the country, that kind of big decisions were made. So genomic surveillance become a very big tool to make a big predictions in the public health. With this, we also know that it also help in the treatment of either cancer or many other disease, even though if we forget about the infectious disease, and it has a lot of scope, which we saw in the recent 20 years, as soon as the SARS coronavirus 1 emerged in 2001, and we saw many other diseases, including pandemic in 2009, HIV is spread, and the reason why we could not develop a vaccine, that again, the changes of the virus, the way it is changing, the monitoring of the this particular notorious virus. Then Ebola, a pandemic, which was to be a pandemic, but it was spread in many countries, caused more than 30,000 deaths. Polio virus transmission, and in the pandemic, we have seen that polio virus has emerged again in many countries, which declared to be eradicated. Zika virus in 2015 and onward in many countries, causing microcephaly, and MERS coronaviruses. So these are some of the high-risk group of pathogen which created panic across the world, and the SARS coronavirus pandemic has a huge impact on today's life. When we talk about the genomic surveillance, there is another aspect of novel unidentified viral diseases discovery, which is a very important fact using this tool. And also, we can sequence not only the virus, but heterogeneous mix of the different genetic material in the same sample, including the host and parasite. So it became a very strong tool across the world. And when we talk about the genomic sequencing, it is not only the virus, but also now it is a deciding factors for the scale of interventions required, what will be the demographic factors which is driving the transmission pattern, and how we can use the diagnostic tool, how fast we can modify them in a way that could become very useful. For example, during the alpha virus introduction, when the variants came, everybody know that there was a changes in spike protein. So if you make a diagnosis tool which is spike based, then may you fail the testing. So those kind of quick information which is based on genomic can change the whole outcome of any outbreak. But at the same time, we have to remember that emerging diseases will always be a reality with the change of climate and the way we are moving from one country to another countries, and a lot of data which has been come across that new diseases are emerging. In 2009, predict a huge network of study was done in 60 countries, and they identified 931 novel viruses across a huge number of the samples from different species of wildlife, livestock, and humans, and which again give an indication that novel viruses will keep emerge. But at the same time, we are also looking at the movement of vector species, a movement of emergence of zoonotic disease because of the change of organizations and other factors, which we all very well aware, and where human also playing a very important role to cause these diseases and outbreaks across the world. And we know how fast we are moving, carrying disease from one place to another place in all over the country. After the pandemic, there is a new term which is now very popular, is emerged in One Health, and recently G20 has Vasudhaiva Kutumbakam kind of logo where we say that One Health is our main agenda, and that include not only the animal but human and also the environmental health, and that cover a broad spectrum, study everything, where you include everything in that umbrella. But when we talk One Health, then also the genomic surveillance become very, very important. And in this aspect, recently the government of India has launched interministerial collaboration is called from the PSA office, One Health Mission. And under that, every importance organization of the country become part of it, and that include from defense to ISMR, DBT, academia, and all the private partners as well. And under that, the One Health priorities has been decided, and slowly the work is also started. And one of the major the thrust area is pandemic preparedness, and also the integrated disease surveillance and joint outbreak response. And whenever we talk all these priorities, we all know that genomic surveillance will be part of it. In 2019, a WHO made a priority list, which is again further under the revision, and further many groups are working for the finding out the priority disease from each family and genus, which can have a potential for coming of any pandemic in the future. And under these criteria, I will look at with the updated Disease X with the COVID-19. At least 4 diseases are present in India, include COVID, CCHF, Nipah, Zika, and these. But if you look at the history of our country from 1957, there were having many high-risk group pathogens present in country, including KFD, Nipah, pandemic influenza, CCHF, Zika, and recently we have also seen the cases of monkeypox, and they have been keep re-emerging time to time. In an ideal sequence of event of any outbreak, the first and most important to identify the pathogen. So based on the pathogen, the containment event can take place, and we can reduce the number of death and spread of the infection. And in this process, we have to anticipate, prevent, prepare, detect, respond, and we have to keep working in the peacetime for the pandemic preparedness. If you look at the data of GSET during the pandemic, the number of sequences coming from 185 member states is a huge data, which is more than 2.5 million sequences across. And there were many countries, they never sequenced during the pandemic. It was forced, politically forced internationally, that they have to do genomic sequencing, because virus was changing in a way, and you need to know which variant is in your country. Recently, we saw the surge in China, where BF7 and other variants were blamed from the Omicron. At the same time, India had no such kind of consequences, though all these circulating variant are also present in country. So genomic epidemiology, phylogenomics, and also the virus diversity of genomic epidemiology become very, very important tool when we would like to know about ancestry of the virus, when virus must have emerged, when it has been transmitted from one country to another country, or to one homogeneous population to another. And even in transmission from a space, if you would like to know about the host movement pattern, immunity, and other factor, then also this data become very, very important. And that's why this particular picture will show that how we require integrated analysis during the outbreak when we talk about understanding outbreak or taking interventions. But also the demographic infections and transmission is also very important in the population dynamics of any infectious disease. The contact tracing data with genomic data become very good and important tool during the COVID pandemic. But also, we also know that there were many super spreading events in many countries, and for that, the genomic data were used to understand and taking prior action same time. If you look at the kind of data which has been emerged in COVID pandemic, there are many other negative impact as well. We don't have storage to hold such data. Many countries don't have bioinformatic people who can analyze all the data in a way that can give a proper output. So data integration of different data sources, the phylogenomic models for bacterial pathogen, host, and also the using of AI tools, which is talk of today, is it become very important, and people are struggling to use that. But at the same time, we also we could develop many other surveillance tool during the pandemic, including feces, sewage samplings, which was used in the past for the polio surveillance. And now even for the monkeypox, many places these tools were used. Historical polio viruses was always detected based on their mutations, and based on the mutation, it was decided which virus is circulating and how many mutations has been accumulated using the traditional Sanger sequencing. But now even polio program has been forced to do the deep sequencing based on the experience of this. So not only the emerging pathogen, but also those diseases which is under the eradication program goes under the scanner of genomic surveillance to know that if we are going to implement any program, either vaccination or therapy, what kind of genomic changes will happen to the virus to escape those kind of eradications program. If we talk about the COVID, the first and important step on the 11th January, when the first genome China released, and based on that genomic sequences, so many vaccines, so many antivirals were made, and based on those vaccines, we could develop the fastest vaccine in the history across the world. Either it is a DNA vaccine, RNA vaccine, messenger RNA vaccine, or vector-based vaccine. So if you look at the history of SARS coronavirus 1, when it happened, it took 3 months to do the genomic sequencing. But now people can do genomic sequencing in a very short time, as less than as even 24 hours, and get the data of a large number of specimens, do the quick analysis, which was not possible in the past. The cost of sequencing is also reduced in the pandemic with the demand of the genomic sequences, and actually that further enhancing the importance and also leading toward this genomic surveillance. Genomic surveillance is now a very much integrated part of One Health program, and everybody from the WHO to different funding agencies has released different funding so that country capacity can be built on this particular platform. In India also, the first 3 cases were immediately sequenced, virus isolations were made, and that really helped in not only making the vaccine, but doing so many other studies, including the detection of variant of concern. When VOCs were introduced in our country, what impact they made on the vaccination program, what impact they made on the breakthrough cases, including later on the mucormycosis and so on. So the story of COVID itself is a big story to talk, but I know that 25% of the resources and manpower was put during the SARS coronavirus pandemic on the genomic surveillance to understand each and everything when virus was mutating very fast from the variant to variant of interest and variant under monitoring, and then finally which are the variant of concern. And in this process, India would also rise not only the co-vaccine, but there are so many vaccines which could be in-house made and they are implemented, and that's why how we could achieve a big target of immunization across the country. In this variant study, we did many studies using our VRDL networks and other network across the country to understand at different time point of entry of these different variants, their isolation and their impact on the vaccine efficacy study. During 2020, in WHO meeting, Dr. Soumya raised these point, which was related to the COVID pandemic, which included the human, what other knowledge gaps that moment, included the human animal interface, clinical consideration, vaccine, and so on. But if I take each and one column from these factors, and if I try to correlate, are the genomic relevance is there? So yes, I can say yes, everywhere the genomic data was required during this pandemic. Either you are doing a pathogenicity study, or you're doing antiviral study, you are trying to develop a drug, or you are trying to decide about the booster. Every country look forward which variant is circulating, what kind of efficacy of the vaccine is there against those variants. So with this emphasis, now everybody across the world in trying to improve, but the India also took a very extraordinary step to establish INSACOG genome surveillance platform, under which 54 laboratories are now functional. And with initially there were a lot of challenges, but finally people came on the platform, and now these platform could be leveraged in future as Ravi sir said, that we can took advantage of these laboratory which is ready for the diagnosis. Similarly, these genomic surveillance network can utilize in the future for different pathogens which is under eradications program, or which is having more important role now in our country to be dealt with. There were many lessons which we learned, and one of the lesson is also we have a great manufacturing hub from the pharma. We have a good INSACOG platform. We have One Health approach, understanding to different stakeholders, and people are understanding the importance of working together, that we cannot fight with diseases working in individual situations. So teamwork. Then also there is an understanding that we require good and good quality of containment facility across the country. But prior to pandemic, do India have not felt the importance of genomic surveillance? So it's not that. The genomic sequencing has been always very important, and it has actually helped in different outbreaks. And I would like to give few of the examples of such an episode. The first is Crimean Congo hemorrhagic fever, which is a high-risk group of pathogen. And actually, initially, it was detected in 2011 in Gujarat, and different episodes of cases were identified time to time. Guidelines were made, and genomic sequencing was done. In the 2011, when we did the genomic sequences from ticks and human, it was shown that there was a reassortment of the virus. And when we did the sero prevalence, we found that 50% animals were having antibody positivity, and the ticks were having the same virus as human. And not only the cases were found in Gujarat, but also in Rajasthan, and also the imported cases from Oman and UAE was reported in India. Genomic sequences help in identify which strain has been imported from outside and which strains are circulating in India. Total 110 complete genomes are present, and India contribute around 25% of these sequences from different outbreaks. We could identify different species, and these data can definitely help in coming future for making vaccine, doing different studies, and also the trials to understand that how these virus strain has impacted and how many percent of mortality they have caused in each outbreak. On similar line, Casanur forest disease, which is a known disease for years from Karnataka state, and has always thought that it is only present in 5 districts of Karnataka. But with the different studies, it was demonstrated that it is present in 5 states, has a wide range of presence this virus. And also we could see its expansion in Maharashtra, Goa. And when we did a study from 1957 to 2017, we could demonstrate that even KFD, a very conserved virus, which is supposed to be having a very low mutation, now has a diversion of new and old KFD strain, which is around 3.2% of the nucleotide changes. But I think all these information help in making a new vaccine, which Karnataka state and these Western Ghats desperately in the need due to lack of good vaccine at present. These data also help in designing the diagnosis and also a different algorithm for testing and which test should be used. So this data and everything is useful, and now we have across the country a level laboratory which is functioning, and they can diagnose the KFD, and you can forward your samples to the respective laboratory across your state, or rather sending to NIV. Similarly, CCHF also has been network has been developed where CCHF diagnosis can be done, and these laboratories has been activated as well. If we talk about the Zika disease, when in 2015, when Brazil reported microcephaly cases, then everybody started thinking it's a new disease to the country. But in 2016, we reported first 3 cases from Gujarat, and sequencing data shown that it was not the strain which was circulating in Brazil and US, but it was a very old strain coming from Malaysia. It means India has this virus from very, very long time. And serological evidence show that India has evidence from 54. And during the pandemic, when we increase our genomic surveillance, we could demonstrate the Zika has spread in now 15 states across the country. So it means that these tools are very, very important. Not only that, Many of the samples also showed the co-infection with dengue and chikungunya infection as well. Many other outbreaks were reported due to active surveillance exist in the country for the Zika from Kerala, from Uttar Pradesh, from Bhopal, and also from the Jaipur state. During the pandemic, we could also detect a case where we reported the first avian influenza case from Haryana, and this case was having a co-infection, and with the very good team here, they could isolate these two different viruses, H5N1 and influenza B, in the same sample, and which caused the death of this child, which was immunocompromised as well. So this also give a new alert to our surveillance system that there is a possibility of having such outbreaks in future. During the pandemic, a lot of alerts has been raised now for avian influenza to be causing a bigger outbreak in human as well. If we talk about the genomic sequencing, we talk about the seaways clinical samples, but now different tools has been developed, which is talking about the air surveillance for the collection of respiratory viruses. And in there is such a good tool that you can send drone, and those devices can pick up the air. You bring them to the laboratory, and then you can sequence the genome plethora from bats, from very remote area where human cannot access, but these drones can go with these machines and pick up your samples. Understanding the importance, there is a ILI, SARI genomic surveillance network has been established, and they have been trained for doing genomic surveillance. At the moment, they are working on COVID and influenza, and I hope that they will be expanding more in their capacity to pick up any new respiratory infection in the future. Recently, monkeypox cases were detected. Genomic sequencing data could show that we in India have two kind of genetic sequencing circulation, one in Delhi and second one in Kerala. All the cases in Kerala is coming from the UAE, and most of the Delhi cases from Nigeria population, and which is having a local transmission going on that particular. We could look at the different mutations, which is very surprising to see a genome like pox is getting very highly mutated, and number of the mutations are observed in the new strains which is circulating in India, UAE, and very few countries. But the B genotype, which is mainly present in Europe, is not present in our country. A similar disease of buffalo pox was having a very huge genome of 220 kb, was detected in 2018, and we could do the genomic sequence. We established this pox surveillance across the country, and because of that, a very rare case of buffalo pox recently detected from Kerala, where the person presented not only with the lesions on the face and hand, but also the genital lesion, which is never reported in case of buffalo pox. And genomic sequences could confirm that it is a similar kind of buffalo pox which is already present in India and have caused such infection. During the Nipah virus investigation in 2001, India could not identify Nipah virus, and it took 5 years to identify the first outbreak. And then we developed a containment facility, we developed the capacity, we have our own virus, and genomic surveillance could lead to identify the 2018 outbreak as fast as possible. During that time, there was a question that the virus is coming from Bangladesh, or it is a bioterrorism act. And then we did genomic sequencing of bats and human samples, and it was showed that it was a new genotype which is present in Kerala, different than Malaysia and also different than Bangladesh, and we called as I genotype. In last 3 outbreak in 2019 and 21, every time you could identify the first case, and further transmission has been stopped. During pandemic, when the Nipah case happened, there were many people have the similar kind of presentation with the cold, cough, fever, and it was a COVID time, huge number of COVID cases in Kerala, but early genomic surveillance and the containment could help in curbing the disease, and no further deaths happen. With this, from the beginning, I was always interested to novel virus discovery. And my first project as a scientist, when I started, I work on many unidentified virus isolates of NIV, and they have been put in right taxonomy places. So if I talk about the past, I used to do the primer working, which used to take 3 months, designing new primer and going through. But in 2017, when I established the genomic platform, in a first go, we could identify 20 new genomes. And I am just giving few examples, including a new species called Kamavati virus, which was called Vardvanani for a long time, and it was identified very quickly. Then we also found a very large number of the isolate of Casodi virus is kept in NIV repository, and it was characterized as a flevovirus. Another virus is called coronaviric virus, was kept for a very long time, and they called a disease in human ARI symptoms. And this was quickly identified using the NGS tool. So in the summary, what I'm trying to say that the genomic surveillance, genomic sequencing is a great tool. But at the same time, if we talk about the outbreak and pandemic, we have to keep doing a pandemic preparedness at different level time to time. India has been doing this, but we need to do more exercise. We have to improve the quality control. We have to work on biosafety, biosecurity across the country. We have a good laboratory. We have very good tempo toward the science research and the diagnostic across the country. Pharma is in very, very good shape to work with us. And with this very positive message, I once again thank you, Dr. Anita ma'am, and the whole team for inviting me to talk. And we always get a lot of inspiration from this inspiring virology team, and we look forward further in future as well. Thank you so much.
Thank you, Dr. Pragya, for that wonderful talk highlighting the role of genomics in pandemic preparedness. We all know that COVID is not the last pandemic we are going to see, and we have a long way to go in pandemic preparedness. Thank you. At this point, I would like to welcome to the symposium our director, Dr. Pratima Murthy, who has joined us despite her busy schedule. I also extend a warm welcome to our registrar, Dr. B.S. Shankar Narayan Rao, who has joined us today for the symposium. I would now like Dr. Anita Desai to talk about her exciting journey at NIMHANS.
Well, good afternoon, and thank you to the management, to our director, Dr. Pragya, and the registrar, as well as all my colleagues here, to putting this event together. And let me also thank the 4 speakers now. You have just got me so excited that I want to put the clock back. Okay, 5 years behind, and so that I can continue my research and collaborate with you wonderful people. Okay, so I'm going to talk about my journey at NIMHANS for the last 31 years as a government employee, and before that also as a student. Now, I strongly believe that in the quote from Theodore Roosevelt, that far and away the best prize that life offers is the chance to work hard at work worth doing. And well, I've been here this long, under the management of 13 directors, okay, 7 registrars, and 3 heads of the department, Dr. Chandramukhi, Dr. Ravi, and Dr. Madhusudan. So I think I've stayed here too long, right?
Lasted it.
So the early years of being a student at NIMHANS, I have this unique distinction of being the very first student in the Department of Neurovirology, Dr. Ravi's first student, and as he just mentioned to you, that I often told him that I taught you how to be a good guide, and he said, I taught you how to be a good student and a guide later, right? Okay. The invaluable guidance that I've received from my mentors, Professor Ravi, Professor Chandramukhi, Professor Gauri Devi, and Dr. Shankar, and Professor Shankar. I've spent a great deal of time in the laboratory alone, working late evenings, because I would start my day at 6:30 in the morning, continue to work till 8:15, then run to the hostel, freshen up, come back, and then start my work on HIV. I would finish reporting at 4:30, take a break for 30 minutes, and get back to my research work. I would work late in the evenings, and many a times, those days we would have frequent power cuts. So walk in the corridor till the power was restored. And the only solace for me in that time, Dr. Shankar Narayan Rao would remember, is the light in Professor Desi Raju's room, which I could see from my corridor. And there was nobody else on that floor, okay, because chemistry was not built Okay, it was just the physiology and was shared with chemistry. And just seeing that tube light burning in Professor Desi Raju's room would make me feel, okay, there's somebody here. At the same time, I knew that far down on the first floor at the far end would be Dr. Shankar. So when I would set up an experiment, put things for incubation, I would go down to Dr. Shankar's room, and those scientific interactions that I would have with him, which were flavored with peanut chickies, was something that I'll cherish, I'll treasure throughout my life. Now, Dr. Shankar had this wonderful ability to challenge young researchers. And one day he challenged me that he said, see, why don't you, I'll give you some 13 brains, okay, of children who have died of JV infection, and they have been, the brains have been collected at partial autopsy. I'll give you 6 regions of those brains. Can you show me if you can locate the antigen in those brains? Well, I didn't know anything about immunocytochemistry at that point, and I decided I'll take up that challenge. So I did it, and I had this wonderful staining, as you can see here, where we even showed a Purkinje cell that gets infected with Japanese encephalitis virus, and which was not shown in the paper that was published by Dr. Richard Johnson on samples that were collected from Vietnam. I got a reprint request from Professor Richard Johnson, who actually coined the word neurovirology. He defined what neurovirology was all about. Okay, when we published this paper in Acta Neuropathologica, I still have that little reprint request, which is signed by him. He was then at the Johns Hopkins University. Professor Richard Johnson and Mrs. Johnson visited us in 1995. Ravi, you will remember this. And this was the old virology before renovation. And you can even see Dr. Madhusudan there with us. I would like to tell you about how I've learned, how I've blossomed in this place, and starting with the services. Now, diagnostic virology. The very first thing that Ravi told me was that diagnostic virology comes first. Research comes next. Right? And so I started off learning clinical virology, testing samples and reporting. The very first HIV ELISA test, which I still remember, designing laboratory tests along with Dr. Ravi's for example, the serological assay for detecting antibodies to measles virus in the CSF, which is a diagnostic test for SSP, which is still today very much revered, and we do get samples from all over the country for this particular test. We collaborated with industry. Ravi has mentioned this to you, with Pat Biotech, with Exciton, and we helped them develop the HIV and JEV kit. And of course, the feather in the cap was the NABL accreditation, for which I must thank Dr. Sandhya Kabra. She actually created this ISO 15189 revolution in the country, and she achieved it so successfully. So we were one of those. We got the National Reference Laboratory and the HIV test and the CD4 accredited in the year 2011. I've also been associated with wonderful national programs, the National AIDS Control Program, right from its inception in 1992, the National Vector-Borne Disease Control Program, the Influenza Surveillance Program, and very recently, the Viral Hepatitis Program, which has been launched in July 2018. When it comes to the national programs, I think NIMANS is the only center, the Department of Neurovirology is the only center which has all the laboratory services. Dr. Ravi was officer in charge till 2010, and I took over from him when he accepted the position of a registrar. So the Integrated Counseling Testing Center, the CD4 Enumeration Laboratory, the Early Infant Diagnosis, which was launched in 2010, HIV viral load in 2012, as well as the National Reference Laboratory. The consortium, you've heard about the consortium of the National Reference Laboratories, the four of them in the country that engage in kit evaluation. We have contributed to national guidelines for diagnosis of HIV testing, as well as the quality systems essentials in the lab. called Laboratory Assessments and Strengthening, which again Dr. Sandhya initiated in the year 209 and in 211. And when I was part of the assessment team in 2009, when I went to these centers all across the country, I just looked at them and I had to ask, why am I talking from a height? I have not done anything for my lab. So I got down to work on ISO 15189 in 2010 and achieved the NABL accreditation in 2011. So when I went in 2011 for assessments, I went with a completely different outlook. I've been a member of the technical resource group for NACO for many, many years now, and we have all have engaged in training for laboratory technicians, for medical officers. And you can see here the first training program, which was done years ago when Dr. S.M. Channabasavana was the director and Dr. Chandramukhi was the head of the Department of Microbiology. The viral hepatitis control program, though we don't have the control program in the MANS, we have still been a part of the team that developed the guidelines and also engaged in master training of master trainers. You can see here one of those pictures where we had this event in Delhi. Now, the most important service we all know that more in the recent times has been the COVID-19 pandemic. And the department has been responsible for testing almost more than 5 lakh samples ever since we started in April 2020. But again, another feather in our cap was getting involved in genomic sequencing for SARS-CoV-2 after we reported the first alpha virus strain, and we identified the alpha virus variant in a mother and child who were returning from the UK on December 25th, 2020. We take a lot of pride in being a member of the INSACOG. We are one of the 10 hub labs, and also with pride we can state that we have contributed 5,258 genomic sequences to the global database. When it comes to research, I've really enjoyed myself. I was appointed as a faculty in February 2002, which also made me eligible then to register students under my guidance. And in the year 2003, Pragya, we can note this, we started with molecular epidemiology. We knew that genetic diversity, population dynamics, and you mentioned phylodynamics today, a new word for me now is that it was very important to do genomic sequencing. And we started that with the rabies virus. I'll come to that. In 2005, I realized that somewhere my heart was also engaged in just understanding how a virus behaves or what its interaction with the host cell. So we started a concept of virus-host cell interactions. In 2010, came in cellular immunology because we realized that HIV-infected individuals with low CD4 counts were still doing very well, and those with normal CD4 counts were sick. So we realized that it was the immune subsets that we had to look in these individuals. The virus vector competence is a study that we took with the Center for Research in Medical Entomology in 2012 to understand virus vector competence. Neurotropism and cytoskeletal alterations that a virus induces was 2014. And of course, the human immune response, which was initiated right from my study in 1990, on Japanese encephalitis. We continued over the years, and we are now engaged with studying the immune response to SARS-CoV-2 vaccine. Just to give a flavor of the research, human immune response for JV, HIV, dengue, and SARS-CoV-2. Molecular epidemiology, we studied the partial sequencing of genes to understand diversity in rabies, HIV-2, and dengue. Whole genome sequencing to understand phylogeny. Molecular biology, we express proteins, the nucleoprotein and glycoprotein of the rabies virus, envelope protein of Japanese encephalitis, as well as HIV-1 and baculoviruses. Point mutations and the effect on virus virulence, a study which I did in the Institute of Virology in Oxford for tick-borne encephalitis virus. Virus-host interaction, protein-protein interactions, identifying host cell receptors, and the cytoskeletal alterations. We studied the vector selection for 3 different species of mosquito, and Also wanted to know how long does the mosquito stay infectious once you inoculate the virus in these species. Cellular immunology, I mentioned to you the immunophenotyping of immune subsets using flow cytometry, which we started with HIV-1 and TB. The first rabies virus whole genome, which we published in 2005, we also dared to clone every small PCR, small and big PCR product that we amplified, with the result that we had clones for the complete negative stranded RNA virus. In fact, the student who worked with me on this particular project, he was asked by his postdoc supervisor if he could bring all those clones together to make to get a full length genome, a negative stranded genome, by doing fusion PCR. And the EF437215 was the first rabies virus whole genome sequence that we submitted, and it had a gene bank name of Nieman's Neurovirology Rabies Human Indian. Receptor identification and protein-protein docking. Once we identified the receptor on the neuronal cells for Japanese encephalitis, we wanted to study how does this particular protein react with the JV envelope protein domain 3. So for that, we used the FT-doc and did stimulations along with the team in Indian Institute of Science. Virus-induced cytoskeletal structures. You can see electron micrographs here of the chikungunya virus budding out of the infected cell. And you can also see that the actin and the tubulin structures, cytoskeleton, which form the cytoskeletal framework, how they react with the virus antigen and what exactly happens to the cytoskeletal structure as the virus infection progresses. Proteomic alterations in infected cells. Here, this is just a flavor, just to show you about how dengue virus, when you infected in human neuroblastoma cells, what does it do and what are the differentially expressed proteins. String analysis to study the interactions further. And of course, the more later, in the last 5 years, we have used advanced molecular techniques and next generation sequencing, metagenomics particularly, to understand what are the viruses in these samples which cannot be identified or detected by the routine serological and molecular methods that we use. So these research projects, I think Ravi and I we are very grateful that we have been able to tap every possible funding agency to get research grants over the years. And it has been a very successful venture, right, for us. Now, training. I have had the pleasure of mentoring. I would say a pleasure, because it is in the whole process of mentoring, you learn and the student learns too. So 10 students as a principal guide, another 10 students who I have mentored as along with the principal guide in the department, either Dr. Ravi, Dr. Madhusudan, and now recently Dr. Manjunath. Where are my students today? How are they doing? This is very important. And I think our director mentioned it the other day, that we need to keep a database on the students who have passed out, who have worked with us and have passed out, to know how are they doing. So my very first student is a professor. In fact, she became a professor almost along with me. Okay. And she's a professor in RIMS Bellary. Nagraj is, I take a lot of pride in how Nagraj worked, because 2.5 years later, when he completed his objectives, he walked up to me, he was a CSR student, and said, I don't want to leave this lab. Can I work for some more time? Okay, so he did work for another time, and he's the one who sequenced the whole genome. Those days it was Sanger sequencing and not something as advanced and sophisticated as next generation. And he is has established himself very successfully in the University of Columbus in Ohio. So Shushmita is in Dallas, Soma is in IISc, Deepashri is in NIH, Montana, Ayushman was with DBT, New Delhi, and now is in Kolkata. He wanted to get back to research and not do more of programmatic work in DBT. Amita is at New York looking out for a postdoc position. Sunita is at the Air Force Hospital in Kanpur. Risha is in New Jersey. And Divya Narayanan, who did her MPhil dissertation with me, went on to study the same topic which she had taken for MPhil here, okay, the site of skeletal alterations. She went on to do this work in the laboratory of a Nobel laureate, Dr. Fazak Ali, in Melbourne, and now she is in Dallas. Anupa is an associate director in Merck Labs in Nebraska. Siddhappa is in Omaha. He's a professor there. Sunil is with Dr. Ravi at Tata MD. Ullas is at NIV Pune. And Vijay Lakshmi and Prachi were at MedGenome. Maria Thomas at Illinois. And Filippo, who was a dual PhD student, is in London, UK. And then I've had the opportunity to mentor MSc and BTech students who worked on their dissertation, and two of these BTech students went on to do their PhD in sunny Buffalo on pharmaceutical sciences. Well, very important thing for me was reaching out. It is all fine working in the laboratory. We do our diagnostic work, we do our research, we publish our papers. But are we really stepping out to see, are there other countries around us who need our help? And this is something which I am very, very happy. And, I'm very grateful that WHO, when they asked us in 2010, right? In 2010, Ravi and me to set up the real-time PCR laboratories. And Ravi decided to go to Bhutan and I was sent to Timor-Leste. I designed their molecular diagnostic lab and later went on to do on-site training for that particular laboratory. The National Health Laboratory in Delhi in Timor-Leste. I've also helped the team in Maldives for Zika surveillance and this revival of cell culture, because it was very important for a national influenza center to revive their cell culture. And that was what WHO requested me to do with the Medical Research Institute in Sri Lanka, as well as the Royal Center for Disease Control in Bhutan. I've helped countries like Bhutan and Myanmar write their national laboratory policy, and Myanmar, Timor-Leste, Maldives, and Nepal to design a strategic and operational plan for laboratory strengthening. Laboratory assessments, Dr. Ravi was also part of this assignment, where we did assessments for Nepal, Timor-Leste, Thailand, and Indonesia. And we've also had the opportunity to work on WHO manuals for establishing virology laboratories in Southeast Asian countries, PCR technology, cell culture techniques, and public health laboratories. And these are some of the manuals that we have done. And this is an assignment that we got in the Department of, so we've taken up the entire agreement of performance of work. Just a flavor of the assignments that I did: the Royal Center for Disease Control in Bhutan in 2014, 16, and 18, Medical Research Institute Sri Lanka in 2015, the Ministry of Health, and as well as the Indira Gandhi Memorial Hospital and the WHO Country Office in Maldives in 2013, 16, and 18, National Health Laboratory in Nepal in 2018 and 19, and the National Public Health Laboratory in Myanmar in 2017 and 2018. This has been a wonderful experience helping these countries build their national policy, build their strategic plans. In fact, I did a workshop on standard operating procedures for clinical and medical laboratories. So I had something like more than 70 or 80 of them in the Department of Biochemistry, Pathology, and microbiology sit together to write SOPs. And this was a work that was conducted for 4 days. Okay, some of my pictures at the different, particularly the one at the Institute of Virology in Oxford, where I spent as a traveling research fellow way back in 1990, between 1998 and 2000, and then continued collaboration with them for another 5 or 6 years. So I'm very grateful to Wellcome Trust because they got my research rolling, and with the money that I got as an International Research Development Award, I was able to fund my first four PhD students. The highs of my journey in NIMHANS, being the first student of the department, I have a great feeling of responsibility. I've still not been able to get over it. Okay. And Detection of HIV antibodies in the very first sample added to the ELISA clade. I can never ever forget that sample. It was referred to us by Dr. R.S. Murthy. His requisition said, psychosis, query HIV. This was a sample which was collected from a lady who was found wandering on Brigade Road, and she was brought to the hospital. Then obviously she was very badly abused. So the sample came to us. That was the very first sample that went into the ELISA clade and turned out to be reactive and was confirmed as positive. The traveling research fellowship from the Wellcome Trust. This was a very important event in my life because I learned molecular biology at the Institute of Virology. I hadn't even seen DNA in a gel before I went, but I had written this grant, sent it for funding, and I went as an independent researcher to this institute. Appeared for the faculty position in Le Mans with an International Research Development Award from the Wellcome Trust, London in 2001. So when I was interviewed in 2001, I had a research grant in my pocket. First department in the institute to achieve NABL accreditation in 2011, introduced new concepts and adopted latest technology with every doctoral thesis. I mentored 10 students as a guide, another 10 as to a joint guide. I never fell short of research funding. In fact, the registrar will remember that I've just sent in an indent yesterday, and I told him that I need those peptides because I want to come in March and complete that work. Okay, I've accepted 18 international and 7 national assignments from WHO. Now a big thank you to all those who nurtured the foundation years, my school teachers, the Sardar Dasu School in Pune, the growing up at the University of Pune, Dr. Apte, Dr. Sardesai, and Dr. Devpurkar, the very first professional steps in virology, Dr. Narendra Singh Dutt, who was a retired dean of IVRI, Izzat Nagar, the blossoming at NIMANS, Professor Ravi, Professor Chandramukhi, Professor Gauri Devi, and Professor Shankar, and at the Institute of Virology, Oxford, Dr. Ernest Gold and Tamara Gritzen. Just a few pictures. I studied in a Parsi school, so you can. My teachers taught me what discipline is all about and what it is to excel in life. Professor Ravi, I can never stop admiring that man, even today, sitting next to this man. Okay? Yes. Okay. Professor Chandramukhi for her generosity. Professor Gauri Devi, I still wear the watchman which you gave me when I was a student. Okay, I'm so sentimentally attached to it. Professor Gauri Devi, I can't tell you enough, ma'am, how there were times when I used to walk into her laboratory, her room, which was on the first floor, somewhere in between the microbiology labs, and her secretary, Mr. Nagaraj, would say, I think you're the only student who dares to go and talk to her, so readily. I once had just completed designing a ELISA for detecting immune complexes to Japanese encephalitis. Dr. Ravi was not around, Dr. Chandramukhi was not around, so I carried the plate to her room to show her that this is an ELISA that is going to work. And she even once told me, when I told her that, why are we sending samples to Japan? Why can't we get those kits here and do the test for HTLV1? Okay. And she looked at me and said, you think you can do that test here? And I said, yes, I can. So you know what she said next? She said, you have all the qualities of your boss, including his arrogance. So that I take it as a compliment. And of course, Professor Shankar for his constructive criticism, for the continuous challenge. He was like a gatekeeper in research. And he taught me what it is like to have good controls in your experiment. And even when I showed it, when that particular slide which I presented, showing the neurons infected with Japanese encephalitis, and said, so that means I've done it. And he said, no, you still have to show that the antibody that you have used is not reacting with 6 regions of collected from the brain of 3 individuals who have died of road accident with no neurological illness. That is the kind of training I have received during my PhD years. I can never forget. And of course, Dr. Tamara Gritzen and Ernie Gold, who taught me what molecular biology is all about. And particularly Tamara, who late evening sitting with a cup of tea, she's a young, pretty, blonde Russian who would sit and talk to me about RNA secondary structures. That is the kind. We lost Tamara 5 years ago, and I think you went away too soon, Tamara. For the collaboration and the funding, Dr. Nivedita Gupta, Dr. Harpreet Kaur, at WHO CRO, Dr. Rajesh Bhatia, Dr. Aparna Singh Shah, Dr. Nalini Ramurthy, Dr. Sirima, and all our WHO country representatives of Bhutan, Nepal, Sri Lanka, Maldives, Myanmar, Thailand, Indonesia, and Timor-Leste for making my stay in those countries so interesting. Dr. Tom Solomon at the University of Liverpool, he continues to remain our collaborator even today, and Dr. Barbara Johnson at CDC and Dr. Jane Bessel at CDC Fort Collins and Kayla at the CDC India office, and Padmini too. She's not at CDC anymore, but Dr. Tyagi, the former director of ICMR CRME Madurai, and Dr. Gopinath at CCRR Bangalore, which is on Double Road, who I met the first time I came to Bangalore. I arrived at his diagnostic lab, and a few days later he said, "You will be wasted in a diagnostic lab. Please go to Nimans." He incidentally was the brother-in-law of Dr. Gopinath, right? Govind Swami.
Yes.
Govind Swami. And to all the administrative departments of Nimans, the purchase, the personal, the claims, the stores, and the accounts. Moments to cherish with my colleagues in the department. The puja after the renovation, you remember, where we had Dr. Shankar and Mrs. Savitri when she was a registrar. The potluck lunches that we have had in the lab, the Independence Day celebrations this year in 2022. I thought I should have a picture to cherish. The Secret Santa that we generally have when we have some, we want to celebrate. Okay, or we just want to enjoy together, and the Ayudha Puja events. Okay. To my parents, for the gift of life and the lessons how to live it. My family and friends, Dr. Purnima Shenoy in particular, who was the first senior resident of the Department of Neurovirology, and she was extremely helpful in my initial days in this institute. And to a delightful child who has grown up into a sensible young man, my son. Now, this is a defining picture of my career. Okay, we have all seen the picture on the left, Dr. Baruch Plumber inaugurating the Department of Neurovirology on the 31st of March, 1986. But I had this wonderful occasion of meeting Dr. Baruch Plumber at the University of Oxford in 2000. And I realized, despite being a Nobel laureate, despite being a chief scientific advisor to NASA at the time when I met him, he was so grounded. He was so simple. He spent 55 minutes with me, telling me all about the stay in Bangalore when he was a C.V. Raman fellow in Sharat Chandra's lab in ISC, and how his wife enjoyed the abstract painting in Lal Bagh. So this is something which I will never forget. Now, I always believe that it is the soil, not the seed. And thank you, Nimans, for making this journey purposeful, meaningful, and so prolific. Thank you.
Thank you, Dr. Anita, for taking us through that truly extraordinary journey of yours at Nimans over the last 3 decades. I'm sure you'll be happy to see a very short video, ma'am. Do we have time?
There is a room full of people waiting downstairs.
So in that case, we will.
I would love to see the video, but I will do it later. But you can go ahead with it.
Yeah, we can have it later. So now I would request our director, Dr. Pratima Murthy, to say a few words. So
as I said, there's a room full of people waiting downstairs. But I just wanted to listen to your whole journey, and as typical as it was. I mean, I think your meticulousness, your attention to detail, your need to document every little aspect of it was really obvious in your presentation, right from your early school days to your high points in your career, and of course, the many milestones in your career. So, I mean, I think it's lovely also to see the intergenerational kind of spread of talent, of dedication, of research, and think it's truly, as you said, it's an inspirational journey, Dr. Anita. And, It's almost like a journey of 1,000 steps begins with the first miles, the first step. And it's almost like these 30 years have been the first step.
Yes, it's always.
Yes, that's what is exciting. Before you know it, life kind of passes you by, your career is over in a wink, in the blink of an eye. But that's, I think, the excitement of being here. So all the very best. I'll have a lot more to say in the next couple of days. Yes. But as I said, I mean, I'm sure you will appreciate that we can't keep people waiting. So I'm going to go. We've Mother Rubbani. You please say what you would like to say.
You have to hand over a moment.
Oh, I'm concerned about that. So all the very best to you, to your family, and to your future endeavors. Yeah.
Ashwin, take one more. Yes.
Can you take from there?
This is all of you join here quickly.
Yeah.
So we'll take picture. Please. Ashwin.
All of us. Oh, sweet.
Thank you.
Oh, he wants to come. He wants to come.
Everyone come.
Varun, yes, sir. Yes, sir.
Thank you. All the very best. And with apologies to all of you, with apologies to all the online viewers as well, and catch up with the video.
Thank you very much. Thank you.
Yeah.
Thank you. I now request our registrar, Dr. B.S. Shankar Narayan Rao, to give a farewell message on this occasion.
So she was my senior. I think most of us, we walked through the corridor, and Professor Ravi, one level of bossism, and Chandramukhi madam in the same floor. For everything, she will scold me. The kind of relationships which we had, the teacher and student relationships, was a great journey which all of us we experienced. Even today also when I meet madam or all the senior mentors whom you mentioned, there's a huge concern which they exhibit, and they groomed us. I think the kind of the composing nature what we have possessed in terms of taking the leadership, running in the forefront, no fear for us. It's all everything, anything for good for the institute, for your department, we are in the forefront. Starting from sweeping and mopping and cleaning the tables and doing the experiments. We never had this hierarchy system or anything. I think both of us, we joined together on the same day as a faculty.
Yes, I remember February.
Both of us, we went together for the medical board and we joined together. And it's almost 4 decades. So I have completed 33 years.
You have completed.
At Nimans as a student, and so we were all students, we were all working together. I think our labs lights were never off. That is the tradition of Nimans. But unfortunately, these years I get to see that more like an office, 9 to 4:30. Yeah, the clock stops, and anytime we can go to Dr. Raju and Dr. Ravi, Dr. Shankar. And Madam Gauri Devi was in the first floor, and Chandramukhi Madam. They are all wonderful teachers, and we never had any inhibition. I think student and teachers, we need not take any permissions. I don't know. So Ravi sir's room, I used to walk all the time. No, I don't think we seek any permissions or anything. That is the kind of relationships. And we, as Dr. Anita mentioned, we all grew with the support and guidance from many of our teachers. That's why we feel very proud. So today we feel very proud when we leave the institute. Our contributions, we feel very proud of ourselves. We talk about it, and we are part of the institute. I think she is one of the persons who had a huge commitment. I think We could have stayed abroad and we could have enjoyed our life, but we had a huge commitment to our country. And NIMHANS gave everything for us. And Dr. Anita, see her contributions initially as a student, taking care of the lab, helping the mentors, working in 2 departments at that point of time, initially when she joined at NIMHANS, and subsequently leadership which she has exhibited, grooming the next generation students and the technical persons. Those are amazing things. And parallelly, the clinical diagnostic services, I will tell you, Professor Ravi sir, he started all this neurovirology from the scratch. The tests and equipments was a difficulty and funds were very limited. But the kind of a cordial relationships and sharing which we exhibited across the departments. In fact, Dr. Raju's microscope and his cell culture work he was doing in virology department. So all of those things which has groomed her, including me, as a scientist. Then subsequently, we are with the shadow of our teachers. They carried forward all our wish lists, helped them. And all our interactions, many times, it was little bit of heated discussions. It was all for the science, actually. At the end of the day, again, we'll feel that, okay, we have learned a lot from our teachers. Amazing research also she contributed. I think Dr. Anita, whenever there is a crisis in this country or elsewhere, Dr. Ravi will be in the front and she will follow. Not only in the country, because all those diagnostic things, which we have to do it, I think NIMHANS is the first and virology is the first in developing the diagnostic tools. The credit should go to the level of commitment has been exhibited by everybody, actually, I will tell you. Starting from attendant till professor, I think everybody has exhibited. Whenever there is a crisis, national crisis, I think, we are all there. And virology, especially Ravi sir. I still remember when we had this COVID pandemic. That's in the month of June. By July, we were starting about the very intense training and also the establishing the laboratories and things like that. By April, we have tasted that heat. That was a very, very intense heat. And then sir was there, I think, near his car. Then I asked him, sir, where are we heading? What is the situation? He said that we should feel very happy. In one's lifetime, we get an opportunity to serve. And COVID has given an opportunity for us to serve. Let us all work. See, imagine that it is the level of commitment. You could have said that, okay, let us wait. But not even a single day waited. And the contribution, diagnostic services. Parallelly grooming the students, the research has paralleled. They're all very cutting edge research. If you look at all those papers, you will get to see that there's not a fragmented papers which we were allowed to publish. They're all fat papers. And overall, contributing to the field as a professional. Neurovirology or diagnostic services. Wherever we go, actually, they ask, actually, who is Professor Ravi, who is Dr. Anita, who is the other faculty in the departments. That means we represent the institute and our work has been identified globally. So, working for 40 years in a department and taking everybody together, contributing significantly. It's only officially you are superannuating today, but our brains and mind is always attached to the NIMHANS. And your boss is sitting here, this is how much he is pushing many things. That is the spirit in which all of us should live. And your contributions in the field and to the department. And overall, we represent the institute. Your contribution is always remembered. So I could see, in fact, and I remember all of you, your students. And how we used to spend a lot of time in the evening corridors,
yes.
sitting outside the library.
Sitting in the and discussing the scooter park, that Dr. Shankar will ask me, now which one are you talking about? Is she there in the scooter park? We took your discussions because they would never end, At 8:30, 9 o'clock, would continue in the scooter park.
Wherever we stand, there the discussion will happen. We don't require a defined area for the discussion. The science happens while you are walking, while you are standing. I'm really, amazed the way your journey. Of course, you require 2 days to speak about it. You made a very crisp. The evolution, actually, all of us, we have evolved over a period of time. And the kind of maturity exhibited by you, I will tell you, amazing. The way, you are pushing the projects and handling so many projects, so many students, so many crises, administrative crises, handling the people will leave and you have to run the lab. It's all we learned over a period of time.
Asking you, can I fast track? Can I just go to the list that we've created as wait list, and especially for the Polar PI Global team?
So, see, there are many people will contribute. I think, as you rightly mentioned, you put Dr. Ravi on the top, because I've seen how closely the department has grew under your leadership. Then subsequently, okay, madam, I will tell you. Madam, and all the directors. So I'm also 13 directors. And of course, the academic director, registrars we had. And of course, Ravi sir is always a fun to work. He was a professor head. He was a dean and COE. And he was a registrar. Imagine his situation, how many things he has to do, and holding international meetings and international collaborations. Amazing, sir. I think all of us, we learned from our teachers.
Grouped by him.
And now if we could work, I will tell you, because I'm working as a registrar, the kind of support I receive from my colleagues, my department, and my students. So science is never stopped. But all of us will be doing so many things to the institute. And the kind of leadership which you have exhibited to carry out the diagnostic services during the crisis. The COVID is the latest one, but I think the NIMHANS virology department was part of all the kind of a crisis which we had, a pandemic situations. So that way we are ever grateful to you. So it's officially we have to superannuate, but our heart and our science is always with NIMHANS, and your mentorship and all whatever you want to share, and then you want to continue the collaborations with NIMHANS. It is always we cherish. And nothing can be replaced with any individual. That's what I experienced. Those vacuums are very much there. I've seen Dr. Shankar, or maybe the different people in our career. There is no replacement which occurs. But that space which we have filled, we feel very proud. We could fill that space. And you did that job. You feel you are proud. We feel very proud we are part of the NIMHANS. NIMHANS always remember your contribution.
Thank you.
So we are grateful to you. I'm sure that the kind of generations of students and the scientists whom you have created, and that mentorship will move faster to the. I think you have 3 generations of students have been done. So several generations. And on behalf of everybody at NIMHANS, I take this opportunity to thank you profusely for your contribution, which is invaluable. Please continue to serve, contribute to NIMHANS. Thank you very much, madam.
Thank you.
Thank you very much, sir, for your message. Shall we have the video? A short video of farewell messages to Dr. Anita from our alumni. Okay.
That is always so cherishing things.
You put them together, is it? Oh,
Volume ಕೊಡು, ಚಂದ್ರು.
Thanks. Cheers. Cheers.
Dr. Desai here. I had a very fruitful tenure as a senior resident during 2004 to 7 under your guidance.
We can't hear.
Ravi sir, when he retired, his student defended 25th thesis. Hello, sir. Yes,
Risha, my student.
just
And
guided.
Ravi was joint guide, right? Risha, no, you're talking about? Yeah.
After that, 26 was Devu.
They do, yeah. Okay. 20, that is for you. But from the department, I'd always had others. When I retire, 25 students should get their PhD.
Desai here,
Please, start from first. Start from first.
senior resident. Hi, ma'am. Dr. Desai here. I had a very fruitful tenure.
Voice is bad.
You want to connect.
Voice.
Cable on connect.
Energy you invested in my
Children,
PhD.
increase, madam.
I fondly remember
Yes, first in
after
the first in the
Hi, ma'am. Dr. Desai here.
Hi.
I had a very fruitful tenure as a senior resident during 2004 to 7 under your guidance. Thank you, ma'am. And I would like to take this opportunity to wish you a happy, healthy, peaceful, and successful retired life. Thank you,
10
ma'am.
years ago, I never could have imagined that I would acquire so much during my PhD. Ma'am, being mentored by you has been a great learning experience. I will always be grateful for the time and energy you invested in my PhD. I fondly remember, after seeing my plaquemates, how excited you used to be. Those encouragements have actually helped me recharge my batteries and work round the clock. Thank you for everything you did to help me improve and grow as a researcher. Hope you enjoy your retirement.
Hello, ma'am. Working with you and being your student was a wonderful period of learning in many ways. You had given me the opportunities, the platform, and all the support to improve me personally and professionally. I loved working with you. I enjoyed the long discussions we had, the life lessons, and the experience you shared. I would like to thank you for all these. Ma'am, I wish you more and more exciting endeavors ahead. Thank you, ma'am.
Hello, I consider myself very fortunate to be a student of Dr. Desai. Ma'am, I really want to thank you for all the learnings. Day by day, I realize that the learnings that I have had during my PhD are more of lessons for life. Thank you, ma'am, once again.
Hello, madam. Wishing you a very happy retirement. Hope your retirement is full of productive and relaxing and fun things. Thank you so much for having taught me over all these years. The most important things that I can think of right now, which are applicable to me all throughout my life, like be it professional or personal, are the importance of dreaming big and the ability to plan meticulously to achieve your dreams, as well as to always lead your life fearlessly. Thank you so much, and wishing you all the very best for all that lies ahead. Bye.
Hi, my name is Nagaraja from New Jersey, USA. I finished my PhD under Professor Anita Desai. Before joining the PhD, I don't know anything about the research. She taught me the basics of research, lab management, and the responsibilities. They helped me in succeeding in my career. She is a kind, patient, and gives the top priority to the students even in busy schedules. Thank you, madam, for your kind support and wish you a happy retirement. Thank you.
Hello, ma'am. I had the privilege of being your first PhD student and have been treasured by your guidance and encouragement. I remember the days you stayed with me late in the evening to train me in classical virology techniques during my PhD days. Your guidance and mentoring gave me a lot of confidence in completing my PhD and now to handle research grants funded by national agency. Thank you for being there. It's hard to believe that you will not be there in virology hereafter. Your students will certainly take virology you taught them further. Praying for your good health and happy days ahead. Thank you.
Hello everyone. Thank you for having me here today. Dr. Anita Desai ma'am was a joint guide for my PhD. As we all know, she's an excellent teacher and a very meticulous researcher. As we're completing a very illustrious career of close to 40 years in Imans today, I wish you a very happy, healthy and productive life post-retirement and hope that your scientific pursuits will certainly continue. A big thank you to you, ma'am, for all the learning and guidance over the years, and I hope that you will continue to enjoy life to its fullest as always.
She's put pictures.
Hi, ma'am. Dr. Desai here. I had a very fruitful tenure as a senior resident during 2000.
Yeah, now we are at the end of the symposium, and it's my privilege to propose the vote of thanks. I thank our director, Dr. Pratima Murthy, for gracing this occasion despite her busy schedule. I also thank our registrar, Dr. B.S. Rao, for honoring us with his presence. We're extremely grateful to all the four speakers, Dr. V. Ravi, Dr. Sandhya Kabra, Dr. Madhuri Thakkar, and Dr. Pragya Yadav, who immediately consented to speak at the symposium when we invited them. We thank them for enlightening us with their insights on various viral infections. We thank all the neurovirology alumni and our faculty colleagues and students from NIMHANS who have joined the symposium online. We also thank all faculty and students from other institutes who have participated in this virtual symposium. We thank the NIMHANS IT section, especially Chandru, for the digital support throughout the symposium. Last but not the least, I thank my colleagues, Manjunath, Ashwini, Varun, Arya, and all staff and students of Neurovirology department, especially Tanmay, Narendra, and Vishal for the help and support in organizing this symposium. Thank you all once again